Hello, welcome to Professor SNAB, where we make A-level biology as simple as a cell. So today we're looking at core practical 9, which is to investigate the antimicrobial properties of plants, including aseptic techniques for the safe handling of bacteria. So the aim of this practical is to investigate the antimicrobial properties of plant extracts by measuring their effect on the growth of bacteria using aseptic techniques to ensure safety and reliability. So for context, some plants produce antimicrobial compounds, and these compounds have the capability to inhibit bacterial growth and kill bacteria. The bacterial growth can be measured using the zone of inhibition, which is something you probably would have explored in GCSE biology already, and that's basically what this practical is going to be about. So aseptic techniques are important as they prevent contamination of cultures and possible spread of potentially harmful bacteria. And it is really important you know this slide because past papers on this particular topic is literally there every single year. So just remember this one. So in this context, it also ensures the validity of results collected and ensures that all so one of the first things you need to do is ensure that all work surfaces are disinfected before and after the practical. Flame sterile the inocculating loops and the forceps. Keep the Petri dish lids closed as much as possible. Please do not leave the lids open. Seal the plates with tape, but do not do it fully as that can encourage anaerobic respiration and harmful bacteria end up growing up. And you do not want that. Carry out incubation with the plates inverted and dispose the cultures in a safe manner. And you'll receive guidance on this from your lab support. So the method, prepare nutrient agar plates, inoculate the agar with bacteria using a sterile inoculating loop, soak the sterile paper discs in different plant extracts, place the discs onto agar using sterile forceps and include a control disc soaked in sterile water. And this is to ensure that the results you collected are reliable. Incubate the plates at 25 degrees Celsius and after 24 to 48 hours, measure the diameter of the zones of inhibition. So from analysis, what you're going to observe is that the independent variable in this practical is the type or concentration of plant extract that you are investigating, and the dependent variable is the diameter of the zone of inhibition. And this is measured in millimeters. The control variables that you will consider in this practical are bacterial species that you use, the agar type, the incubation temperature and time, and the volume of extract on the discs. Larger zones of inhibition demonstrates greater antimicrobial activity, and no zone demonstrates that there are no antimicrobial effects. So some evaluation points that you can observe is that there are some limitations in this practical. So for example, the diffusion rate of the compounds may differ, the concentration of the extract may vary, measuring the zones can be subjective. So your idea of the diameter could be different from someone else. Use of only one type of bacterial strain is also another limitation, as you can get a completely different set of results with a completely different bacterial strain. So some improvements you can do for your practical are to use replicates and test a range of concentrations. Use digital calipers for accuracy. So this is to do with measuring the diameter of the inhibition zone, and compare the results and observations between different bacterial strains. So what are the different safety considerations that you need to ensure during this practical? So incubate at 25 degrees to reduce the growth of pathogens. Never open the plates after incubation, and this is something you need to remember. Use PPE during the entire practical and ensure proper disposal of agar plates. And again, the guidance on disposal is likely to be given by your lab support. So that is all from Professor SNAB today. Hope you found this video useful. If you have any questions, let me know in the comments below and I'll get back to them as soon as possible. And don't forget to like, share, and subscribe.